Good afternoon. My name is Jared Posh and I'm with Codex DNA. Welcome to today's SLAS Transformed tutorial. Today's tutorial is called Biotherapeutics Accelerated, generating genes, gene variants, and clones on your lab bench using the BioXp 3200 automated system. My name is Jared Posh and I am your host and tutor today. I'm a field application scientist with Codex DNA. Thank you for joining us. Today's agenda will be a company overview, as well as what is synthetic biology, followed by introducing the bioexperience, an overview of the BioXp 3200 automated system, where I will go into more detail about gene fragments, clones, and variant library builds, from design to transfection-ready DNA with our new RapidAmp feature. I'll cover a few case studies and some potential applications, and then finally a summary. Our company originally grew as a division of synthetic genomics, the SGI and dsDNA, which was our former name, focused on the development and commercialization of unique and proprietary technologies that enable synthetic biology from Synthetic Genomics Incorporated and the J. Craig Venter Institute. We spun out in 2019 as a new and unique corporation with the BioXp 3200 system, Gibson Assembly reagents, variant library services, VMAX cells, and more. We are now focused exclusively on empowering researchers to rapidly and independently code and write synthetic DNA. SGI DNA is now Codex DNA. This rebrand took place because we, as an organization, needed a name that reflects where our company is going rather than where we've been. Why Codex? A codex is an ancient manuscript, and there is no manuscript more ancient and more meaningful than the code written into the DNA of every living thing. Just as software developers use code to build applications, Codex DNA is helping scientists code DNA to build biology. Our Chief Technology Officer, Dan Gibson, views building biology from an engineering perspective. Dan and his team like to take DNA apart and put it back together again to understand how it works. The team can edit and program DNA just like coders program a computer, but their apps are very different as they program life. This approach is known as synthetic biology. Dan and his team invented Gibson Assembly as part of their mission to create synthetic cells and to map the essential genes responsible for life, known as the minimal cell, published in 2016. In 2010, his team came up with the first synthetic flu vaccine, which gave rise to 2013 commercialization of the BioXp, the first DNA printer, which we will be talking about later in this presentation. But this system enables benchtop DNA synthesis and cloning. This became the foundation for the digital biological converter concept known as the DBC, which is a prototype that is envisioned to enable rapid vaccine development in response to global pandemic events, enabling the code needed to create medicine to be shared between treatment centers globally over the internet. In 2020, our company was the first to assemble the first fully synthetic SARS-CoV-2 genome. We made this genome available to the scientific community to assist in rapid vaccine development as well as positive controls for a diagnostic test. So let's dive in a little bit more on our automated system, BioXp 3200. The BioXp 3200 system is the first and only push-button automated platform for building DNA fragments, clones, and libraries. It is a fully automated synthetic biology workstation that allows every scientist to discover, design, and rapidly build high-quality target genes, essentially hands-free, overnight, starting with electronically transmitted sequence data. These systems synthesize highly accurate DNA fragments and use Gibson assembly for the final DNA assembly of the fragment into the customer-supplied vector backbone, or expression vector. Its unique error correction enzyme allows for the synthesis of highly accurate DNA fragments, clones, and libraries. So how is BioXp revolutionizing custom DNA synthesis? Well, it allows you to take control of your workflow and your intellectual property. This gives you the peace of mind knowing that your vectors never leave your lab. Also, you can rapidly iterate your DNA constructs. This allows you the freedom from tedious cloning workflows, allows you to design, build, and clone, and even amplify your DNA overnight, up to 32 at a time. Elucidating gene functions and pathways are another key benefit of the BioXp system. It allows you to complete projects in days instead of months. So how do you get your DNA on the BioXp? Well, it's pretty straightforward. You go onto our website and create an account on our ordering portal, and then you submit your sequences online. From there, our complexity checker will give you immediate feedback, and you have the option to codon optimize to remove complexities or any issues with building that DNA. Once everything is set to go, you place your order, and you will receive your BioXp kit in 3 to 5 days. This includes all of the reagents already pre-dispensed, including the error correction enzymes and the purification reagents. Simply load the system, which takes just a few minutes and is very straightforward, and then press start. There's no software that you need to operate, no methods you need to write. Everything is already built into the system. After the overnight reaction, you retrieve your DNA, and your DNA will be ready for transformation or direct transfection in the case of RapidAmp. This is a menu of the things that you can build on the BioXp. You can build fragments, you can clone those fragments into expression vectors, or you can build variant libraries. So the key benefit to the BioXp is that you can supply your own vector. That vector might have trade secret or proprietary information, or it may be licensed. Regardless, it never leaves your laboratory, and the only thing that we need to know are the Gibson homology ends. Gene library variants can be made either in a scanning format or in a combinatorial format, which will be discussed later, but a key feature are the degenerate bases. All right, I've received my order. Now what? This is what the kit looks like when you receive it. You'll receive 2 components, a 4-degree component and a -20 component. Simply store everything in the appropriate refrigerator freezer, and when it comes time, you thaw and load them into the instrument. There will be an empty provided PCR strip in the case of cloning where you can dispense your linearized expression vector. Simply add 12.5 mLs of ethanol, close the lid, and press start. It's very straightforward, and a checklist is provided with every kit. Let's take a closer look at each mode on the BioXp, starting with gene fragments. Gene fragments are purified double-stranded DNA fragments ready to use in all downstream processes. You can clone these fragments using Gibson assembly or the cloning method of your choice. The sequence guidelines are 300 to 1800 base pairs, and the GC content can range from 20% all the way up to 70%. The product specifications include 32 reactions per run, 1.5 micrograms per reaction in TE, high fidelity error correction, and these are made in an overnight reactions that takes around 16 hours. The next mode is BioXp cloning mode. These can be either single fragment or multi-fragment clones. The clones are made from fragments synthesized de novo on the BioXp. You synthesize first and clone second. You can either match up one fragment per one clone, or you can do multiple fragments into a single clone. We call that multi-fragment assembly. You can also provide more than one vector. In fact, you can have up to 4 different unique vectors in a single automated run. The cloning adds 2 hours additional processing time, and the output would be 20 microliters of clone product ready for transformation. There's only a small amount of the fragment used to make the transformation ready Gibson assembly product, so the unused fragments are available for additional downstream steps. A key feature of the multi-fragment clones would be antibody engineering, specifically single-chain variable fragments. This allows you to make larger constructs or have additional constant regions that can include a hinge or a linker, or even an additional enhancer and promoter to drive 2 different chains at different expression levels. These can be both cloned or uncloned, and it allows you to assemble 2, 3, or even 4 de novo pieces into a single construct, enabling a massive amount of flexibility. And the final mode, which builds on the previous modes, is the RapidAmp amplification workflow. This allows you to take the clone that you built in the previous step, where you assemble 1 or more de novo fragments and or constant regions into an expression vector, to amplify that plasmid up to about a 10 microgram total quantity using a rolling circle reaction that we have optimized significantly. It's random hexamer based, and it takes an additional 4 hours. Once that's finished, simply take the plate off the instrument, do a quick restriction enzyme digest, and you will have linearized versions of your plasmid that are transfection ready, free of endotoxin. This allows you to amplify genes that are toxic, or allows you to preview phenotypes prior to performing glycerol stalks and DNA plasmid prep. A key feature of RapidAmp is that it eliminates the transformation growing and prepping, while also providing high yield. This allows you to significantly consolidate the workflow down to a work week. It allows you to generate 4 times or more the amount of data than you can through the traditional method, and also, again, allows you to have endotoxin-free preps in about a week. And last but certainly not least, and the mode you've probably been waiting for, libraries. Libraries on the BioXp can be either scanning libraries where you replace 1 or 2 or 3 codon positions at a time and then scan them along the protein so that you can test systematically the function and role that each amino acid is playing in the overall phenotype of a protein. And then finally, once you've identified those positions, either through scanning libraries or bioinformatically, degenerate libraries. So this allows you to target very specific positions so that you can interrogate the function of the protein with every possible combination of specific amino acids at defined locations. Variant libraries are also a version of degenerate libraries where we can reduce and isolate down the codons to very specific ones instead of a degenerate pool. Scanning libraries look something like this typically. You scan 1 or multiple codons at a time and then shift to the next codon and on to the next. These are no different than a typical run on the BioXp. You can scan 32 positions in a run, and you can get 1 or multiple BioXps building these at the same time. Combinatorial libraries are a bit different. All of the different possibilities are going to be pooled into a single tube. Sometimes we need to make replicates of these to generate enough material to go directly into yeast or a genome integration strategy of your choice using homologous recombination. Regardless, in this case we're showing 560,000 possible variants, but we can make millions, in fact approaching billions of different variants all in the BioXp. With so many possibilities in a single reaction pool, it's really important that you only have diversity in the selected locations and not in the locations where you are not intending to have diversity, because that would complicate things even further. So as you can see in the figure below, with top 100 reads, you see no errors outside of the regions selected for diversity. This indicates a high quality combinatorial gene variant library. So that's the end of the libraries portion, and just to wrap things up, the BioXp system and its advantages. Again, fast turnaround time at low cost and low labor cost. Efficient high-quality DNA builds in a hands-free overnight run. Enhancements and application expansion via software updates and reagent kits. And robust application development across a wide range of genomic workflows, whether we're talking about genes, molecular tools such as positive controls or vaccine development components, genetic elements such as enhancers, promoters, you can bank those with the BioXp. You can go straight to protein production, and we have something exciting that I'm going to talk about next for that. Antibody libraries, which we just covered, and cell line engineering. So in addition to BioXp, we offer a wide range of catalog reagents, such as the Gibson Assembly Hi-Fi Kit or Ultra Kit. Please contact us for a sample if you source these from other vendors so you can compare the quality of our kits side by side. But Gibson Assembly Ultra is exclusive to Codex DNA. It allows you to clone a wider range of fragments at higher efficiency. Codex DNA also offers the RapidAmp technology that I referred to a few minutes ago with the BioXp as a standalone kit. This reagent kit allows you to combine both Gibson Ultra and RapidAmp in a single master mix reaction. And finally, when it comes to protein production, definitely give Vmax a try. Vmax is an alternative to E. coli. The name of the specimen is called Vibrio nitrogens. It allows you to grow your protein quickly. In fact, it's one of the fastest growing bacteria known. It grows to higher ODs, and the protein will have higher solubility. It allows you to express a large amount of protein in a faster workflow. Thank you for your attention, and now I will open up for a live Q&A session. Thank you for your time.